Hello my dear students and welcome back to Excellence Batch, and we have successfully completed the chapter Human Health and Diseases so far. It's time to start with another important chapter, that is biotechnology, principle and processes. If you are a NEET aspirant, this chapter is really important for you. If you are CUET aspirant or even the CBSE board aspirant, obviously you have to give the exams, right, board exam, then again this chapter is very important because this chapter or this entire unit, biotechnology, which comprise of two chapters, the principle and processes and its application, they both are very important, and from here great questions can be formed. So that's why they have more weightage in any of the exam. Now biotechnology, most of you must have heard of it, and whenever you think of biotechnology you start thinking of sci-fi movies and all, and in the movies you see they made some virus and that virus went through the city, people dies and so, right? So that's one part that's a movie. But if we talk about this field, biotechnology, this is a great field. This is a field for research. This is a field for teaching. This is a field for doing, work in the industry. And so, if you are not into becoming a doctor and you want to explore some other fields of science, and you are really interested in technology and research at the same time, must try biotechnology. So there are different types of degree in biotechnology. Some is biotechnology honors, some is simple biotechnology, some is biotechnology engineering as well, where you need mathematics also. So these fields are very, very nice fields nowadays. You can work with the pharmaceutical industries, right? You can work with the production lines, you can work with the chemical industries, you can work with the food industry, you can work with the vaccine, you can work in vaccine production, and you can work in a lot of things, right? Antibiotic production and so all that comprise of biotechnology. So see, now I have told you, in a lot of fields biotechnology has its application. So if you think that you don't want to be a doctor, you can give that a shot in your life, right? And those who think that doing MBBS and become doctor is everything, and if it does not occur to you, then it's the end of the life. It's not. If you are interested in this field, you definitely can excel in it, and when you are interested, you can obviously do better, you can excel, and you can earn better, and you are happy doing that work. At the end of the day, you are very happy in life. So it's nothing like this field is the lower than MBBS. It's not like that. It's all in our head. The only thing is that whatever you're doing, that should be of your interest, not anyone else's. So because if you have interest, you will be very curious to know about things, and you can study well, you can invest your time, you can invest your energy, and you will be happy. So anyways, let's talk about this biotechnology, principle and processes. So today we are just talking about the introduction part. First, what is a biotechnology, right? So if in a layman's language, don't just go on these words first. In a layman's language, if we see what is biotechnology, let's split the words. Here we have word biotechnology, right? Okay, let's change the color. Here we have the word biotechnology, right? So bio means living, and technology is technology. So we are combining biology and technology, engineering, right? So let's see what will happen when you join both, and why are we doing so. See, the organisms in the entire world, they're capable of a lot of things. Let's take an example of one very simple bacteria, lactobacillus, right? LAB, lactobacillus bacteria, or lactic acid bacteria, right? So it is present in the curd. So what power does this bacteria have? The power this bacteria have is it can convert your milk into curd. If you'll add the culture of this bacteria into a boiling milk, what your mother used to make curd at home, you must have seen. So when in the boiling milk we add a little culture of the curd, that's the culture of bacteria, and that in the morning, if you keep it at night, that milk will be converted into curd. So what we are doing with biotechnology, we are using these same organisms. What organism? Organism here is this bacteria. We are using this organism, we are changing them a little. We are playing with their genes, we are playing with their DNA, we are changing them a little so that whatever function they are performing, whatever products they are producing, we can get them in a better way and in a more efficient way. Fine. So there is one federation, the European Federation of Biotechnology. They have given the definition, complete definition of biotechnology. According to them, it's the integration of natural science and organism. That means you're taking the natural science, the living science, the biology. Okay, and the organism, cells, parts thereof, and molecular analog for product and services. Now, in that natural science, whatever organisms we have, like we have this bacteria or any cell we have, let's take an example, the beta cell of pancreas. What does beta cells of pancreas produce? Insulin. Okay. Or any product these cells are producing, or molecular analogs. That means any molecule that are similar to that particular product. For example, if your beta cells are producing insulin, so we have one exact particular thing that is equivalent to that insulin. So that's a molecular analog, or we can say the artificial form of that. Okay, for products and services. And why are we using that? So that we can make a product out of it, and then we can provide service to the human beings who need it. Let me give you a very simple example. So we are using natural science biology, right? In biology we study a disease, that is diabetes. So what happened in diabetes? The diabetic patient is not able to produce insulin effectively. Why? What if we integrate science and we use beta cells of pancreas and we somehow modify it and we culture the cells and they will start producing the insulin. Now insulin is what? The product, right? What are we using? The cell. Okay, and that insulin will then be sent to the all human people who have diabetes and they will be cured, and that's the services. So what basically we are doing is we are using our biology, the study of the living organism. We are using the organisms, either their parts or their products or their molecular analogs, so that we can extract the product out of it and we can then use for our human welfare. This is what products and services means. Okay, that's the complete definition of this particular the definition that is given by the EFB, European Federation of Biotechnology. And make sure you should also learn this, who have given the definition and what is their full form. Sometimes they simply ask you the full form and you there get confused. What's the full form? European Federation of Biotechnology. Now what are the products so far we have made? We'll be discussing that side by side. Okay, all right. Now if we talk about biotechnology, it is of two types. Yes, the biotechnology has been divided into two types. One is a traditional biotechnology. It's a conventional biotechnology. What is conventional or traditional? That means it has been used by human like since time immemorial. From ancient times we are using it. Then at that time we didn't know there is a biotechnology thing, right? But we have been using it since long time. Like if your mother or anyone in your house who is doing household work and they don't know, they have never read biology in their life, they will never know what biotechnology is. But still they are using biotechnology every day. How? They are producing curd from milk. Or when you make dosa or idli, we ferment the batter of dosa for around 6 hours, right? So that is also biotechnology. But our grandmothers, they didn't know what is biotechnology, but they were doing it. So that's a traditional or conventional biotechnology which has been used since ancient time. Since ancient time. Right. So in this one, basically we are not playing with the organism, we are letting organism play. You understand? We are not manipulating genes, we are not using any technology, we are not taking the organism to lab. We are just using the natural powers, natural abilities of the organism to do what they are doing. For example, lactobacillus bacteria is in the curd. You never know it's there until or unless you have read it in your science class. right? So until then we didn't know. So people also didn't know that it's due to a bacteria, so they were letting the bacteria play. So traditional biotechnology is all about not disturbing the organism, rather the organism will produce the product for you by its own natural abilities. So let me give you some example. The example is like curd making, vinegar, cheese making. That's happening like much time ago. Baking, in some baking we use yeast for fermentation. Dosa making, right? And alcohol like beer they make. So all these things are the example of traditional biotechnology. Genes are not manipulated here. Genes are not manipulated. It is based on organisms' natural abilities. Organisms' natural ability. So whatsoever it is already doing, it's doing. Right? Second is a modern biotechnology. In modern biotechnology, now we know the definition of biotechnology and also we know the techniques how to do it. So in modern biotechnology, we are basically manipulate the gene. We play with the genes because now we know what is a DNA. Now we know what DNA does. Now we know what gene does. And this is how we can change the genes. Okay. So in modern biotechnology, the genes are manipulated. You can add the gene, you can subtract the gene, and somehow you can, involve a lot of technology with it. So here technology is involved. Technology is involved. Here technology was not involved. The traditional one was more of the art rather than the science. But this is more of a technology in science, right? So with modern biotechnology, we have produced like DNA vaccines, right? The vaccine for hepatitis B that I've told you we make, right? And also here we have made humulin. Humulin is the insulin that is produced in bacteria and we are using it to cure diabetes in many patients, right? And other than that, what have we made with the biotechnology? A lot of things we have made. Test tube baby is the perfect example. Test tube baby, right, etc. A lot of things are there in modern biotechnology that we have made so far, and making therapeutics and all, everything is involved. So here two technologies are the one without which the modern biotechnology is incomplete, or we can say the modern biotechnology have two major techniques. One is genetic engineering. So whenever someone is opting for modern biotechnology over the traditional, they have to do two techniques, or they have to process through two techniques. What is genetic engineering, and second is bioprocess engineering. Without this, the modern biotechnology is incomplete, or it has no value. Now what are these two engineering? Let's see. So first is genetic engineering. In genetic engineering, what we do, we are manipulating the genes. So when we manipulate the gene, we are manipulating their genotype. What's a genotype? For example, if you have done the chapter genetics, in genetics, you must have heard of some factors or genes which are involved in your particular trait, to express particular trait. For example, this is our DNA. Okay. So in DNA, we have one segment of DNA known as a gene. Imagine this is a gene. Okay. Now this gene has two alternative forms. For example, this is one gene for height of individual. So the gene have two alternative form, capital T and small t. Small t, capital T, capital T. For example, if there is one person who has taller height, the person might be having this or this trait. Okay, so the person will be having tall height. So this is its genotype, it's present in the genes, right? And this is its phenotype, that means what type of a character it is showing. For example, my height is not that tall and it is not that small also, so somehow treat me in a smaller people. Okay, and imagine I have like shorter height, if I have shorter height. So that means my genes have this kind of allele, that is my genotype. Now what is my phenotype? The shorter height. Okay. So what if we use biotechnology, what if we use biotechnology and change my gene from TT to capital T? Then my height will be taller. I will be very tall. Okay, so this is what we do in genetic engineering. We change the genotype, we change the gene of a person. What is a gene? It's a piece of DNA. What if I cut the DNA and put the right DNA? Then my phenotype will also be changed. My phenotype will also be changed. So read here. Technique to alter the chemistry of genetic material. That means what's a genetic material? DNA. I am changing the genetic material to introduce these into the host organism. And who's the host? The organism in which you are expressing. Like in our example, we have taken my body. Okay. And thus change the phenotype of the host organism. And now the character is changed. Let me give you one more example. For example, this is a human beta cell. This is the human beta cell. Okay. The beta cell have a DNA and that DNA have a gene. Okay. Now this gene will produce a protein and that is insulin. That is insulin. Okay, now we want that insulin should be produced, but we cannot have a lot of beta cell. So what we did, we took a gene out of it. The gene which was producing the insulin, we took it out. And we added the gene into a bacteria. Added in a bacteria. Because now bacteria have this gene that produces insulin. What bacteria will do? Bacteria produces insulin. So what we did, we changed the genetic material, right? And by that the phenotype of the bacteria changed. What's the phenotype? Production of a protein is also a phenotype. What it is expressing, it's a phenotype. Okay, so bacteria produces insulin. So what you see from outside is a phenotype. And what it expresses is also phenotype. But genotype is what type of gene it carries. So what we do, we alter the chemistry. How we alter the chemistry? We change the DNA. When we'll change the DNA, gene will be changed. That means genotype is changed. And when genotype is changed, phenotype will definitely be changed. Because whatsoever your genotype is, that decides the phenotype. Okay, phenotype is what you see externally or what it is expressing, and genotype is what type of a gene it has. And what's a gene? It's just a segment of DNA. Okay, all right. And these capital T, small t, we are giving in the sense to make it easy, because I can't explain you the entire sequence of DNA. So one sequence of DNA, like it is A, T, 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 they give it the capital T name to make it more easier. Okay, so one is genetic engineering, where we are playing with the gene, putting it here, there, and doing so, cutting it, putting it there, changing the phenotype. And along with that, we need another engineering, that is bioprocess engineering. Now what is this? So where are we performing all these things? In the lab. In lab, everything is microscopic, at microscopic level, right? So we have petri dishes, we have microscope, we are doing it here, right? Now imagine you're performing this experiment where you are taking the insulin gene and putting it into the bacteria, right? For your sake, you are doing it in bacteria and you feed the bacteria with the medium, agar medium. Now imagine if that lab is not completely clean and it is with a lot of dust particles and contamination. So after few days you will see the bacteria that you have grown, it does not contain any bacteria, rather it has a lot of fungus. Why? Because the entire environment was so much contaminated. The plates you have used, the petri dishes or the equipments you have used, they were also contaminated. So as a result, what happened? The fungus from those contaminated products or anything, they enter into the medium, they contaminated entire culture. So is it a win-win or a fall situation? It's a fall situation for you, right? So that's, here come the another chemical engineering, which is a bioprocess engineering, which is all dedicated to how can you make the entire environment or the lab or whatever equipments you are using contamination free. That means without any microbes, that means sterile. So this involves maintenance of sterile ambience in chemical engineering process to enable growth of only the desired microbe or eukaryotic cell. If I want my bacteria to grow, only my bacteria will grow. The fungus should not grow. In large quantities for the manufacture of biotechnological products like vaccine, enzyme, antibiotic, etc. And what's the product here we have? The product we have here is insulin. So without these two, your biotechnology is never going to happen. It's a waste of time, right? So these are the two core techniques of biotechnology. First, genetic engineering, where you are actually doing a work, you are cutting the genes, changing the phenotype, and so. And here you are maintaining a sterile condition, the contamination free condition, so that only desired organisms should grow that you want them to grow. Okay, so let's talk about the gene cloning. So now we are going to talk about how genetic engineering is done, and how genetic engineering is done, that is best explained by gene cloning. What is gene? The segment of DNA. Cloning is to make the exact copies of it. Exact copies. Just like a Xerox copies. Okay. For example, you have a gene for which, for what? For insulin production. Now, how can you make millions of copies of that gene? That's a gene cloning. Okay, so for doing this, the important step of a important technique of genetic engineering, we use a technology known as RDT, known as, okay, rDNA technology. R means recombinant, also known as recombinant DNA technology. So that's a technique that we use when we want to make the exact copy of a gene. Now, how is it done? Let's get started with that. So how gene cloning is done? Let's see. It's a very wonderful and most interesting mechanism here. You will understand how recombinant DNA technology is done. Okay, before starting this, why recombinant DNA technology is known as a recombinant DNA technology? Okay, technology you all understand because it's a tech. Recombinant DNA. Because in recombinant DNA technology, we form recombinant DNA. Ma'am, okay, but what's recombinant DNA? Whenever a foreign DNA is added to the main DNA, for example, this is a main DNA, that's a main DNA, okay, that's a main DNA of any organism. When you cut the main DNA, make a place. When you cut the main DNA, make a room, make a place and add a foreign DNA to it. So this green one is a foreign DNA. Now the DNA that has been produced is now be known as recombinant DNA, because this has been formed by the combination of two types of DNA, the main DNA and the foreign DNA. Okay, for example, this is the DNA of a bacteria, and in the DNA of bacteria we have added the gene for insulin. So it's a foreign DNA, right? So now this DNA is considered as recombinant DNA. So in recombinant DNA technology, we produce recombinant DNA, and this technology is very important whenever we are performing gene cloning or we are making the copies of a gene. Okay, so for example, you want to make the copy of a foreign DNA. So definitely you need to make the recombinant DNA for it. Now let's discuss this in much detail, and that will make more sense to you then. So in gene cloning, what we do, first of all, we take a vector. A vector can be anything. Like here, the vector is a plasmid. You all know what's a plasmid? You must have done it in the bacteria. It's extra chromosomal, double-stranded DNA present in the bacteria and in some yeast, right? So we always take a vector. Vector are like vehicles. Like a car. Car helps you to transport from one place to another. The vector is also needed to transport a desired DNA, a foreign DNA, right? So for example, here we have a main chromosome of human. It's a main chromosome. So from main chromosome, first of all, I will isolate a desired DNA, which is a foreign DNA. Okay, so I have a lot of DNA, right? Out of that DNA, there is one gene that is producing insulin. Imagine the desired gene that I want to take out is the insulin gene. How I will cut this desired gene or foreign DNA, or also considered as a DNA template, from the entire main chromosome of us with the help of molecular scissors known as enzyme. For example, this desired gene, let me just paint it with the other color. Imagine this desired gene is somewhere present here. I want to cut it and then put it separate. Then I will be using one enzyme known as restriction enzyme. RE means restriction enzyme. RE means restriction enzyme. Restriction enzymes are also known as molecular scissors, which we'll be discussing later on in detail. The function of these molecular scissors is to cut the DNA segments in between, right? So I will be cutting the desired gene from my main chromosome with the help of these enzymes. Similarly, because I want to add this desired gene into the vector, only then the vector is a vehicle and it will move, right? For example, if in the car you want to go to your school from your house, then there must be a space in the car to sit, right? If there is no space in the car, how will you go? You will not be able to go. So what we do, we also make space here. We also cut this with the help of restriction enzyme. Let's cut it. So this is how it is. Now we will join these both, and what will be formed? A recombinant DNA. So what will be formed here? Recombinant DNA. So this recombinant DNA is formed how? By joining the vector and the desired gene. And how will you join it? For joining we need again an enzyme, that is ligase. Ligase is also known as molecular glue. This was cutting, this is joining. Just like in art and craft class, what are the two basic things that you need? You need a piece of glaze paper, or that any paper, nowadays you use colored papers. You need a scissor and you need glue, just like we used to do in art and craft class, right? If you don't have any of the thing, you will not be able to make anything. So remember, that piece of paper is your DNA, the scissor is a restriction enzyme, and the glue is your ligase. So I am joining these two DNA with the help of enzyme ligase here, right? Now the recombinant DNA is formed. So this vehicle is ready. For example, this is a car, space has been formed, you are a desired gene, you have already taken your place in the car. Now where the car will go? In the host organism. So the host organism, for example, we have a bacteria which is E. coli. This is E. coli bacteria. E. coli bacteria have its main chromosome also, Escherichia coli, right? And now, because plasmids are the part of bacteria only, now this plasmid, which is the vector, it can easily go inside the E. coli, right? Like this. Alright. Now what will happen? This E. coli will multiply. So when this E. coli will multiply, along with its multiplication, the desired DNA will also multiply. Imagine this E. coli, I am making it a little shorter. It has made its copies like this. Okay, so when it has made its copies, everything will multiply along with that. So the foreign DNA will also multiply, and this is how you have made copies, number of copies of your desired gene. You have made number of copies of your desired gene. Right, very simple. You have taken your desired gene which you want to make copies of. You have taken a vector or vehicle, you put it into that, you made a recombinant DNA. Now that will enter inside the host organism, and host is the E. coli. Now whenever we are adding the foreign DNA with the vector, we make sure, where are we aligning it? We will always link it where ori is present. What is present? Ori. You understand what is ori? Imagine ori, or origin of replication, is present just right here. What is origin of replication? It is that segment of DNA from where the DNA replication start. And how will the copies will be formed? The copies are forming by DNA replication only. It's a gene, it's a DNA's part. How will it get multiplied? There is no Xerox machine. They have to multiply with the help of DNA polymerase enzyme by DNA replication. So whenever you have to make sure that DNA replication should definitely take place, the desired DNA should definitely be joined with the ori, origin of replication. Okay, so what we did? taken the desired gene, put it into the vector, made the recombinant DNA. Once it is made, we put that into the host. Now host will multiply, and when the host will multiply, it will produce a lot of copies, and along with that the copies of desired gene will also be made. So basically it is involving the techniques of formation of recombinant DNA, hence recombinant DNA technology, and gene transfer. So gene transfer is very important step, is a very important step in our DNA technology in gene cloning. Okay, now one more thing. When the vector enter inside the host, we also call this process as transformation. So the process when the vector is entering inside the host, what you call it as transformation. So if someone asks you what is transformation, when vector or recombinant DNA enters into host, you call that process as transformation. So the entering of a vector or a recombinant DNA into the host is known as the transformation. And now because it has taken the vector, this host will become a transformed host. This has become a transformed host. What is a transformed host? The host which has done the process of transformation successfully, and it has taken the recombinant DNA into it. Okay, all right, or the taken the vector into it. All right, so this is how the gene cloning is done. So this for the first time anyone must have done in their life, right? So for example, there are always people who are first to go on moon, first to fly, first to do this and that. Just like that, who were those first scientists who performed gene cloning for the very first time? And their name is Stanley Cohen and Herbert Boyer. These were two scientists who performed the first RDT in 1972. They performed the first rDNA technology experiment. They are the first, they are the pioneer, right? So, all this rDNA technology was possible because of one important thing, the discovery of restriction enzyme. Imagine there were no restriction enzyme. Then you will not be able to cut the DNA. You will not be able to cut the DNA. Now, what they did? So, first of all, what they did, they took a vector. And the vector in this case again is plasmid. Vectors are what? Vectors are vehicles. What they did, other thing, they took an antibiotic resistant gene. Antibiotic resistant gene from a bacteria which is Salmonella. Alright, so to clear out all this, let's first discuss what are antibiotic resistant gene. These are the genes which will confirm antibiotic resistance in bacteria. For example, if I have one bacteria, this bacteria have a main chromosome and this bacteria also have plasmid. In the plasmid of bacteria, these genes are present which are antibiotic resistant gene. That makes this particular bacteria resistant to antibiotics. For example, I gave this bacteria antibiotic X. Now this bacteria is smart. With evolution, it has made this gene in it, right? So this bacteria will not be killed with this antibiotic. Okay, so antibiotic resistant bacteria are those bacteria which have antibiotic resistant gene. That means these gene will produce certain protein which will protect the bacteria against certain antibiotics, right? So if any bacteria have antibiotic resistant gene, the bacteria is antibiotic resistant. That means that bacteria will not be killed by any kind of antibiotic or a particular type of antibiotic, because these genes are also particular to the antibiotics. Okay, so what are antibiotic resistant bacteria? Antibiotic resistant bacteria have antibiotic resistant genes. which protects them from killing by antibiotics. I hope you all know what are antibiotics. These are also certain proteins which are secreted by bacteria to kill the bacteria. Yes, secreted by bacteria to kill the bacteria. Okay, now what these scientists did, they took a bacteria Salmonella typhimurium. What is this bacteria? Salmonella typhimurium that causes typhoid, and they took this gene which is antibiotic resistant gene out of its plasmid, and now they took the another plasmid with no antibiotic resistant gene, with no, or that means antibiotic resistant gene is absent. Okay, now how did they have taken this gene out? Again, from the plasmid they have taken this gene out with the help of usage of which enzyme? Restriction endonuclease. Just like there I have told you, there's a main chromosome and from that you have taken the desired gene. So here antibiotic resistant gene is your desired gene, and that vector is a plasmid with no antibiotic resistant gene. Again we did same with this one also. We have to make the space. We again use molecular scissor, the restriction enzyme. We have made the space. Let's add them together with the help of enzyme ligase. So we use the ligase enzyme, and what will be formed here? A recombinant DNA will be formed. A recombinant DNA will be formed. So this is a recombinant DNA. Again, we will be adding this gene. We will be adding this gene near the origin of replication. So what these two scientists did, they took the gene, antibiotic resistant gene from antibiotic resistant bacteria. They took this plasmid from that bacteria which was not confirming antibiotic resistant, or that was sensitive to the antibiotic. That means if this bacteria from where we have taken this plasmid, if we will feed that with antibiotic, the bacteria will got killed because it does not have antibiotic resistant gene. Now what we did, that's a recombinant DNA we have formed, recombinant DNA that we have formed. Now we will do the transformation. How we did the transformation? And what is transformation? Putting the recombinant DNA or vector into the host. And what's the host here? The host is again E. coli. Host is E. coli here. Now what we did? We put this recombinant DNA into the E. coli. Now what E. coli will show? E. coli, they are not antibiotic resistant bacteria somewhere, right? So this strain of E. coli was antibiotic sensitive, right? That means it can be killed by antibiotic. So now when it has antibiotic resistant gene in it, now E. coli has been converted into antibiotic resistant. Antibiotic resistant. Okay, so this is how, just by adding one gene, by doing gene transfer technique in gene cloning, they have made E. coli bacteria from being antibiotic sensitive to antibiotic resistant, right? So this was the first experiment of recombinant DNA technology which was performed by these two scientists, the Cohen and Boyer, in 1972. Fine. Okay. So when there was no biotechnology, what were the traditional methods that were used? The traditional method that were used was traditional hybridization, right? And how biotechnology is better than that? Yes, you know, in traditional hybridization technique, okay, let me give you an example here. How biotechnology is better than traditional techniques? Traditional hybridization. So there is one technique that we are following from the time immemorial, hybridization. This is one technique we are following since time immemorial. What we do, we take one female, we take one male, superior male, like we take a superior bull and we take a superior cow, yes, and we cross them, and the babies are produced. Okay, but this traditional technique does not involve technology. So both these male and female we use are of superior quality. But is it always the case that the babies they are producing will always be of superior quality? No. Let me give you a simple example. So sometimes the father is taller, the mother is shorter in height. So we think father is taller, the baby will be taller also. But then the baby came out in the world, grow in size, in later in stage, the baby's height is shorter, not taller. So somehow you tried, yes, the father is taller, so baby will be taller. So it's not the case. So this is like hit and trial method. You will never get the result. But how the biotechnology is kind of a boom here. What we will do, for example, if we want to make a superior babies, what will we do? We'll take the ova, manipulate the gene, right? We take the sperm, we'll manipulate the gene. Now this manipulated gametes with better genes, they are crossed, and whatever baby they will be formed, definitely it will be of superior quality. Why? Because we have changed the genes. We are not waiting for the traditional techniques crossing over to give you that, right? So we have made it deliberately. So that's it's always said that these techniques are better than that, but they are not performed in humans, because there are some ethical issues, otherwise we'll produce something that will be not under our control. So there are certain ethics in biotechnology also. There is a there is someone who is watching what you're doing, and there is some stop points that where you need to stop in research, otherwise we'll make something that will be not under our control. Okay, so that's about traditional, hybridization, and biotechnology. Let's see what are the 3 basic steps of genetic engineering. Let's sum this everything up which we have done so far. There are 3 basic steps in genetic engineering. First, we'll identify which gene do we want to insert. Like in the previous one, I've told you the desired gene can be an insulin gene, or the desired gene, for example, in the Herbert Boyer's experiment, it was this antibiotic resistant gene. Okay, so first step is identify which gene you want to study or you want to work with. Second, introduction of the identified DNA into the host. Now, after making the recombinant DNA, how will you put that into the host? Okay, how will you introduce that into the host? How will you do the transformation? That's the second step. Third is maintenance of introduced DNA into the host and transfer of the DNA into progeny. And the last is their cloning. Yes, the host has been has taken the recombinant DNA, but now will it be able to get maintained there? And after how many generations it will remain there? And will it multiply or not? That's the third step. So within 3 total steps, your genetic engineering is summed up. So that's about the entire introduction part of this chapter. Let's solve, first of all, let's see this diagram and then we'll solve some question. Okay, that's a diagram from NCERT. It's given in restriction enzyme. Okay, so that's a foreign DNA, that's a vector. Okay, now some restriction enzyme cutting both foreign DNA and vector DNA at specific point. So at some point from where you want to cut the DNA, it will be cut by restriction enzyme. Alright, that has been cut. Now they both have been joined. You can see the desired DNA has been added here. So this DNA molecule is recombinant. Now how have we joined it? With the help of ligase, which is a molecular glue. Now we have done transformation where the recombinant DNA has been entered into host and host will divide and your desired DNA will divide. So that's a more beautiful form. Mine was not more beautiful. So let's solve some question. Which of the following is not the part of modern biotechnology? Test tube baby, DNA vaccine, humulin, vinegar making. In modern biotechnology, we have test tube baby, DNA vaccine and humulin. Humulin is human insulin, which has been produced by our DNA technology. But vinegar making is in traditional biotechnology. It's a part of traditional biotechnology. Okay, so answer to this question is 4. Next. Which of the following are the techniques of genetic engineering? Okay, gene transfer, gene cloning, RDNA technology, all of these. So in genetic engineering, what are we doing in genetic engineering? Ma'am, we do nothing because we don't know how to do it. Now you know it. We alter the chemistry, we alter DNA. When we alter DNA, we alter genotype. That means we have changed the DNA, that means genes has been changed. And by that, their phenotype will be changed. So by doing also, we need some techniques, or for doing also, we need some technique. First, you have to transfer gene, you have to multiply gene, and how? By making recombinant DNA. So answer is all of these four. Next, arrange the steps of genetic engineering in correct sequence. A. Introduction of the identified DNA into the host. Identification of DNA with desirable genes. Maintenance of introduced DNA in the host and transfer of the DNA into progeny. So first thing that you do is you identify what DNA you want to work with. So first is B. Second is then you will make sure it will go into the host by making recombinant DNA. Then is A. And then you will multiply it in the host and maintain its progeny. So answer is B, A, C, third. This in this sequence the steps should be. Okay. All right. So that's about the introduction part of biotechnology. We have just introduced you biotechnology, what it is, how it is done, but this was all the theory part. In the next one, we'll be talking about the enzymes in detail, and later on, I'll tell you how it's done in the laboratory. And it's going to be really interesting. So keep on studying biotechnology with me and study it nicely. Read the NCERT, and I will meet you in the next class. Till then, bye-bye. Work hard.